Hello and welcome. My name is Alice Driesen and I'm presenting the poster Detecting Cancer Cells in Acute Myeloid Leukemia using a Variational Autoencoder. First, a short introduction of AML or Acute Myeloid Leukemia, which is a cancer of the bone marrow that occurs in a myeloid lineage, which you can see here on the slide, especially in the branch that leads in the end to the monocytes. During AML, somewhere along this developmental tree, the cells start expanding rapidly and you get a big expansion of a certain cell type, which is then called AML. During normal developmental trajectory, the cells in the bone marrow change their surface markers as they develop and as they differentiate. So we can use these surface markers to distinguish between the different cell types. However, during AML, it's difficult to use these cell surface markers to recognize the cancer cells. First of all, because there's a huge heterogeneity between patients and even between the leukemia of the patient before and after treatment. Secondly, because the healthy cells go along this developmental trajectory, they change the surface markers constantly, and it's difficult to distinguish aberrant expression from a cell that's just transitioning from one stage to another and is not quite in either one of the stages yet. So in short, it's difficult to identify the cancer cells, to identify where along the developmental trajectory the cancer occurs, to get the cancer-associated markers or patterns, which in the end is all very important if you want to look for new therapy strategies for AML. We have this data set from a pediatric AML study that we want to use to investigate AML a bit further to hope to overcome some of these difficulties. This data set, we have flow cytometry of 23 surface markers of in total 20 patients. Here on the left you see the 23 surface markers that were fed through UMAP, so you see the first two UMAP dimensions in a top colored by these 20 patients. In the bottom, we colored them by the time points. For most patients, we have a diagnosis and a relapse sample in blue and orange. And of only 4 patients, we have a remission sample, which you see in green. You already see that's not that many cells compared to the diagnosis and relapse. And these remission samples, they're indicated with EOI. We consider them healthy because it's a full remission sample. Using this data set, we want to use a variational autoencoder to kind of solve 2 problems. First, to distinguish the cancer cells from the healthy cells, and secondly, to find out where among the developmental trajectory does the leukemia occur. In order to do this, we train a variational autoencoder on only healthy cells. The input will be the 23 markers, and we train it only using the healthy cells, and then the autoencoder will first encode these 23 markers into a latent space with a lower dimension than the 23 input markers, and then decode this again into the full 23 dimensions of the markers that were inputted. So if your autoencoder works perfectly, your input and your output will be exactly the same. And since we train it only on healthy cells, we basically select it and train it to be very good in reconstructing a healthy cell. So if you give it a healthy cell, it should be really good in reconstructing it. So after training and we set all the weights and we select the model that best reconstructs the healthy cells, we can give it some non-healthy data that we get from the relapse samples and the diagnosis samples. And these samples are, depending a bit on which sample you choose, a mixture between healthy and non-healthy cells, because it contains both cancer cells and healthy cells. And what we hope is that the cells from this sample, they will split a bit, so you will have a very good reconstruction error for these healthy cells, so the input and output will have very little difference, and it will be a really bad reconstruction for the cancer cells because they're not like the healthy cells, so the model will not be that good in reconstructing them. Then what would be really nice is if this latent space, which is like a reduced dimension already, still captures this developmental trajectory of cells, and we can kind of use this to find out where among this trajectory these cancer cells fall and which cells are close and similar. So first we looked at the mean squared reconstruction error to see if we can use this to distinguish non-healthy cells from healthy cells. In total we trained 400 auto-encoders with different parameter settings and on different folds of the healthy data. And you see here a summary of these 400 models. The test and the validation sets are then folds of the healthy cells or the healthy data set, and this non-healthy is a combination of some samples that we know had a very high percentage of blasts. So most of these samples were non-healthy cells or cancer cells. And you can really see that the average mean squared reconstruction error of this set, which contains a lot of cancer cells, is higher than for the samples and the cells that have no cancer cells. So we can really use this mean squared reconstruction error to distinguish between cancer cells and non-cancer cells. The second thing we looked at was the latent space of the model. And in this case I'm showing you the latent space of the best performing model. And we chose the best performing model on the mean squared reconstruction error of a healthy independent test set. So there was the best model for reconstructing healthy cells. And because this best model had a latent space of 6 dimensions, we did a UMAP on these 6 dimensions to be able to visualize it in 2D. And you see in the UMAP really that in the top right corner you have the immature cells, which are orange. Everything that ends with a P is a progenitor cell, and it goes along this circle to the more mature cells, which are the mature monocytes. So it's really nice to see that the latent space of this variational autoencoder still captures this developmental trajectory that we know is true. This UMAP shows all the healthy cells, so all the remission cells. And what is extra nice is that we can map or encode using the same model a non-healthy sample of a patient, and then also show it on the same UMAP, and that is what you see here on the right. So here on the top you see cells from a diagnosis sample of a patient, and they fall mostly in the immature cells, so mostly among the progenitor cells. But if you now look at the relapse cells of this same patients, you see that they fall a bit further among the developmental trajectory. So the relapse occurs in a more mature developmental stage than the diagnosis. This relapse is more along the promonocyte or monoblast stage. So we can really capture where among this developmental trajectory we find the leukemia of different patients. In the future, we would like to include a cell type classification loss so that this autoencoder can also, from the latent space, predict the cell type, and we hope that this will improve the latent space so that this trajectory in the latent space become even more clear. And what we also want to do is annotate some of the clusters from the diagnosis and relapse samples, so from the non-healthy cells, so that we have some cells about which we're very certain that they're cancer cells. And then we can make some synthetic mixtures of cells that we know what percentage of this sample is cancer cells and what percentage is healthy, and then see how some of the metrics that we're investigating behave. And that would also, if we have this annotation, would also help us to get a quantitative evaluation of how well this approach works and how well it would be in classifying cancer versus non-cancer cells. With that, I would like to thank you for your attention. I would like to thank my co-authors for their collaboration and their contributions, and I hope to see you at our posters. Thank you.